Hi, my name is Jess, and I spent my summer at the University of Colorado Boulder's Change Lab studying psychology and neuroscience. So the main study that I worked on was called the FORCE study, and it focused on the effects of exercise on elderly adults in their everyday lives. So to enter the study, they had to be at least 60 years old, and they had to pass an exercise test, which is the picture to the left, where we measure their max heart rate just to make sure that they're physically capable of taking it. And then they go through a series of fMRIs and cognitive tests, and if all that goes well, they begin their exercise. So they come in 3 times a week for 16 weeks for a half an hour, during which, this is the part that I got to do, we measure their heart rate, their perceived physical exertion, which is how hard they think they're working, and their speed and their grade. And then when all that is done, they go back through the cognitive tests, which I actually got to participate in, because while I was there, they were training a new professional research assistant. So I got to do the cognitive tests that usually the participants get to do, and they were actually really hard, so that was really cool. And then the other half of what I did was in what they call the wet lab. So it's in the same building, but it's just where they do all the blood protocols. So everything, that's me wearing gloves, you have to wear gloves because everything is considered contaminated because it is real blood. So the main thing that we did was centrifuge blood into plasma and hematocrit, so that's the centrifuge in the middle. And then we take the plasma and we tested for cannabinoids, which is just the level of cannabis in your blood, because the studies done at the Change Lab are mostly about cannabis. And then we took whole blood and we added infection to them to test the level of cytokines, which is your ability to fight infection. And then the rest of the blood work was DNA and RNA extraction. So for the DNA extraction, it's this whole long process, and you end up with these little white strands of DNA that you can actually see and that we can actually measure. And then we send those out to Denver for further analysis, just because it's still a part of the cannabis study and we didn't have that capability. And then we also got to do RNA extraction, which I got to take part in, with aliquoting plasma and putting it in different tubes and storing it in the freezer, which is kept at negative 80 degrees Celsius, which is really cold if you don't know Celsius. We had to wear gloves to even open it, to put stuff in it, and it beeps at you after like 10 seconds of having it open, just because it has to be kept so cold. And you can't actually see the RNA when it's done, but there's a machine that it goes into that laser measures the amount of RNA so we can quantify it and write it down. And I also got to do data entry, so I entered all the exercise data and a lot of the blood work data. And then I spent a couple days working on the fMRI, so that's an fMRI machine, and the participant lays down and they put their head in the cage, and then they look at a mirror, which is reflecting a projection on the back of the fMRI, which is cognitive tests. So they do cognitive tests while they're in the machine that we administered from the control room, so we can see their answers and their results. So the fMRI takes videos of their brain, essentially, while they're either lying still, answering questions, looking at photos, taking cognitive tests, and stuff like that. And then the most surprising thing for me was how meticulous it was to do the blood protocols. Everything has to be done in a biosafety cabinet like that, even the DNA and RNA extraction. And so everything that goes in there has to be sprayed down with ethanol before it goes in there. And if it's in the DNA and RNA cabinet, it has to be sprayed with RNase away, so to get rid of any extra proteins. And it's really small, so you have to lift your elbows up in there to pipette and stuff, and it's really hard. On the first day I was working in the wet lab, the undergrad that I was working with took the blood out of the centrifuge so it was fully separated, and he punched the cabinet on accident and it mixed it. So we had to re-centrifuge it, and then he took it out again and he dropped it and it remixed it, so we had to do it again. And it ended up wasting about an hour of our time. I stayed at work an hour late that day just because we have to get it done, and you only get like one sample from each participant. Like, you can't go back and be like, give me more blood. And then we also had to test one of the professional research assistants' blood because we got new red blood cell lysis and we were out of it and we didn't know if it worked. So instead of asking for participants' blood, we drew like each other's blood. Like, I got to watch and we just tested that. And then the things I did for fun, I had a super awesome host family with 3 little kids, so I hung out with them a lot, and I went to my host brother's baseball games, and we went on hikes, and I also went on hikes by myself because they can't hike very far because they're very young. So I hiked around Boulder, and then there was also 3 other Pinterns in Boulder as well, doing different things. And so we went to the movies, and we went bowling, and we hung out on Pearl Street, and we actually went to Elitch's, which was really fun. So that's us eating snow cones at Elitch's. And then my advice for future Pinterns is to take every opportunity that's given to you, because there's going to be so many given to you in the lab, outside of the lab. And it's such a great experience to learn about yourself and to learn about the area you're interested in. So just take everything that's offered to you. Do everything you can while you're there. It's a short time, and it's really awesome. And here's my thanks to everyone who helped get me there.
Yeah.